Focusing light on the retina. To see things clear, we need to focus the light on the retina, especially in the area of fovea centralis. When light enters our eye, it goes through the cornea, then aqueous humor, then pupil of the eye, to the lens, vitreous humor, and the retina. First it goes to the neural layer of retina and photoreceptors inside of it. And then light turns into electrical energy, and action potential goes to the brain through the optic nerve. So please note this pathway of light entering the eye. At the cornea, light is refracted, changes its direction a little bit. It's also refracted when it enters the lens and exits the lens. And our lens can change its shape, its curvature a little bit to allow the light to be focused on the retina. For distant vision, how our eyes adjust. We do not need to adjust our eyes for distant vision, because distant rays of the objects that are located 20 feet or more away from our eye, they go parallel, they enter the eye, and they do not require any changes from the lens. Ciliary muscles are relaxed. That's the easiest for our eye to see things 20 feet or more away. For close vision, our eye needs to make 3 adjustments. First, accommodation. Lens should change its shape, and ciliary muscles from all the sides, they press on the lens, making it more round. We partially lose that ability to accommodation over age of 50, and that condition is called presbyopia. When people over 50 cannot see clear subjects that are close to their eyes. Another accommodation is constriction. Pupil of the eye becomes smaller. Pupillary reflex constricts the pupil. And finally, conversion. It's medial rotation of the eyeballs. Here it is. You can see example of conversion. That nice cute kitty looking at the object, a fly, very close to his eyes. Okay, now problems of refraction that so many people have nowadays. Nearsightedness or myopia. Here it is, myopia. When light rays concentrated in the retina, in their eyeball, in front of the retina, instead of being on the retina like it happens in the normal eye. So nearsightedness can be corrected with concave lens. Hyperopia is the opposite problem, farsightedness, when light rays are concentrated behind the retina, and it's corrected with a convex lens. To see how it works for normal eye and what happens in the case of hyperopia and myopia, go to YouTube and see this video. How the eye works, animation, how do we see video, nearsighted and farsighted human eye anatomy. You also can copy this link from the PowerPoint. Astigmatism is another problem. It's caused of unequal curvatures of the different parts of cornea and lens, when cornea or lens are not smooth enough. It also can be corrected with different types of lenses. Let's talk about photoreceptors right now. So light enters the cornea, goes to the pigment layer and is reflected, and then goes onto photoreceptor cells. We have cones and we have rods. And each of them, both of them, have outer segment, here it is, and inner segment. Inside of those cells, there are special visual pigments or photopigments. Those are molecules that change their shape as they absorb light. Here's one of those molecules in the rods. Rods we have for dim light or for white and black light. So rhodopsin, here it is, the whole molecule is called rhodopsin. Inside it's made of 2 proteins. The large one is opsin, and the smaller one in the center is retinal. This retinal molecule can be detached and attached back again to the opsin, and it's synthesized from vitamin A. So how it works? In the dark, rhodopsin forms, so retinal is attached to opsin with help of vitamin A. So we need vitamin A to see well in the dark, in the dim light. Sometimes some people see well in the bright light, but they don't have good night vision, and the main reason of that would be just lack of vitamin A in their diet. When bright light comes in, rhodopsin breaks down and retinal is detached from opsin, separates. When it happens, it causes action potential that travels through that cell and then through the neurons into the brain. So excitation of cones is similar to excitation of rods, but we won't talk in details about those molecules. You need to know that there are three types of cones for blue light, green, and red light, and all other colors we are seeing when there is activation of more than one type of cone at the same time. So combination of those colors. For example, combination of blue and red give us violet color. Color blindness happens when a person cannot see one or more colors. It happens because of the genetic condition, and it can be passed to next generations. Most common type of color blindness is when they cannot see the difference between red and green colors, and most often it happens in men. Light adaptation. When we are moving from darkness into bright light, when you enter the dark room, turn on the light. First of all, you cannot see well. It requires adjustment from your eyes. Pupils constrict, retinal sensitivity change, rod function ceases, and after several minutes you start seeing better, but full adaptation happens in over 5-10 minutes. We also have dark adaptation when we go from bright light into darkness, and this process takes more time, usually within 20 to 30 minutes. You also need to know visual pathways, but let's do together from the very beginning. Light enters the eye. It goes first through the cornea, then aqueous humor, then lens, then vitreous humor, then it goes to the cornea. From the cornea it goes on inside of the cornea it goes on photoreceptor cells, then through the neurons of ganglion cells it goes through the optic nerves and goes to the place next to hypothalamus, which is called optic chiasm. In optic chiasm, 50% of those nerve fibers keep going on the same side of the brain, and 50% of the fibers cross on the other side. So that is decussation at the optic chiasma. Why is it necessary? So both parts of our brain could know the things that each of our eyes is seeing. Okay, so after optic chiasma crosses fibers, signals go to the thalamus, and another neurons carry information to the occipital lobes, where we have visual cortex. So visual cortex in the occipital lobes. That is your visual pathways that you need to know.